Thank you, everybody, for coming. It's my pleasure to introduce Piotr Witkowski, who's coming from the University of Chicago. He did his medical training in Poland, in Gdansk, and then, as we were talking about last night, kind of, I guess by accident, ended up at a transplant meeting about a year later. Ended up in Columbia University in New York, and really never left. He did his surgical fellowship training at Columbia, and then thank you, everybody, for coming. It's my pleasure to introduce Piotr Witkowski, who's coming from the University of Chicago. He did his medical training in Poland, in Gdansk, and then, as we were talking about last night, kind of, I guess by accident, ended up at a transplant meeting about a year later. Ended up in Columbia University in New York, and really never left. He did his surgical fellowship training at Columbia, and then made his way to University of Chicago, where he's now a professor of surgery, and is the director of their pancreas and islet cell programs. Just as some of you might know, islet cell transplantation, unlike Canada, is not a funded activity. It's typically not paid by insurance or by the government. So it's quite challenging to be able to do islet cell transplant. And to Piotr's credit, they have really been able to build really a world-renowned islet cell transplant program in Chicago, and is one of the busiest centers, definitely the busiest center in the US, but one of the busiest centers in the world. And again, to his credit, they've played a really major role in a lot of the clinical advances in islet cell transplantation in the last decade, either as leads of several clinical trials, are definitely playing a huge major part in those trials. So it's our pleasure to welcome him, and we will look forward to your talk today.
Thank you very much. Thank you very much. Good morning. I appreciate the introduction, very kind introduction. So as we know, and people ask me, we're not quite there to offer islet transplant to every type 1 diabetics, and insulin remains the standard of care for most patients. And especially now with the closed loop system, the medical care is improved. However, even the best closed loop system, they sense and release insulin every few minutes, where islets are sensing and releasing insulin every millisecond, right? So the glucose control, that's why the glucose control with the islet is still much better than with the best technology. So yes, we have the technology and closed loops, but we still have patients who, despite the best technology, the best education, the best efforts, they struggle. They have really poor glucose control. And these are the patients which we try to help with the transplant. So as we know, pancreas transplant in the United States, it's a standard of care for those patients. However, as we know, it's a major surgery, has risk of complications, risk of thrombosis, and still around 10% risk of bleeding and operation, in addition to immunosuppression. So that's why we offer pancreas transplant to those patients who need a kidney transplant, they need a surgery and immunosuppression anywhere, or those highly selected patients with only type 1 with good kidney function, who have hypoglycemia unawareness, and then because of this, they develop severe hypoglycemic episodes, which are life-threatening. those patients live in constant fear of sudden death, that they can die during the night. So those patients, we can offer pancreas transplant. So islet transplant was developed as a minimally invasive alternative to whole pancreas. We take the pancreas from the donor exactly the same way as for whole organ transplant. Then we take the pancreas to our facility, we isolate the islets, and then we infuse them in the portal vein through the catheter place. In many different ways, but one most common way is through the catheter place through the skin by interventional radiologists into the portal vein. This is our facility. We are lucky to have GMP facility, clean room at University of Chicago, and to control the environment, to provide sterile environment and sterility of the islets. So quickly about islet isolation, we inject collagenase, the enzyme, through the pancreatic duct into the pancreas, is dispersed, and then we cut into pieces. We put in the special container. It's called Ricordi chamber. Dr. Ricordi developed the system. And then we warm the collagenase. Collagenase dissolve the collagen, and then exocrine and endocrine tissue is being released, and then we collect this tissue. So basically, it's liquid pancreas. From single pancreas, we collect between 20 or 60 ml of tissue. And of course, we cannot infuse so much tissue into the portal vein. It could cause thrombosis. So what we do, we are purifying, separating exocrine tissue from endocrine tissue. So we end up with between 2 to 5 ml of tissue, purified islets, which we then infuse into the portal vein. It's not much surgery here. I love it. I just massaging the back. That's all we do during the islets infusion. So a patient typically is just under mild sedation, can watch the islets and dripping. So about the immunosuppression. So historically, the breakthrough was in 2001, Edmonton group, they modified immunosuppression, islet isolation. And so they proposed steroids-free immunosuppression. It was induction with taclizumab. Now we don't have it, but we have basiliximab. And then immunosuppression, they propose using rapamycin and low level of tacrolimus. Over the time, the insulin, so patients got insulin independent, most of them. However, only 7% of patients remain insulin-free 5 years later. So the conclusion was that this immunosuppression might be not sufficient enough. So therefore, not strong enough. Therefore, we start using induction with thymoglobulin or different T depletion agent. And then in different studies, we showed that outcomes are much better. So the conclusion was that T depletion induction improved the immunoprotection, and this is still necessary for the islets, and 5-year insulin independence increased to 40-50%. However, okay. So as Trevor mentioned, in the United States, we don't have the reimbursement. So 10 years ago, we were at the edge of closing the programs, the funding expired, but then I was able to join European multi-center trial, where when we were tested, and we were the only center in the States to be part of this trial. It was randomized trial, when in addition to regular immunosuppression, we've been testing reparixin versus placebo, and reparixin, it was the new agent, anti-inflammatory agent. It inhibits leukocytes migration to the site of the transplant. The idea is to decrease the inflammation around the transplant for 7 days, improving the islets engraftment. So in this trial, the steroids were not allowed, just to see the full anti-inflammatory effect. And when we didn't use any steroids and we gave thymo, our patient developed cytokine release syndrome, and then they had primary islets loss. So this was lesson from me, that the thymo without steroids, it's not the best solution at the same time. So in order to increase chance for success, we spaced the dose of the thymo. The dose was 6 milligrams per kilo, the regular dose. We spaced it over 8 days, so giving half of the dose per day over 18 hours, just to minimize all the risk of the cytokine release. So the red dot, it's a primary non-function. So in the placebo group, okay, this graph presents the islets engraftment, which we measure based on the islet function on day 75 during the mixed meal tolerance test based on the C-peptide secretion and glucose control. So the islets engraftment for placebo, as you can see here, but in Reparixin, we saw substantially improved engraftment comparing to placebo. So in my cohort of patients, Reparixin has a major impact on islets engraftment. However, it was multi-center trial and the outcomes were very different in different centers, and overall conclusion was that Reparixin makes no difference. However, there were so many variables. We had, in our cohort, it looks like there was a major impact. The drug was manufactured by Dampé in Italy, and it was never approved for this application. So it's not available, basically. So what we did after the trial, we couldn't use Reparixin anymore. So we went back to anti-TNF. Everybody has been using anti-TNF, Enbrel. And I start giving the Thymo 1 month before the islet transplant to avoid the risk of the cytokine release and the primary non-function. So in subsequent patients, as you can see, the engraftment was not really improved comparing to placebo with Enbrel, but at least we avoided primary non-function and cytokine release syndrome and allergic reaction to Thymo, which require steroids, which are not the best for the islets engraftment. So for me, the conclusion was Thymo is great in the long term as immunoprotective agent and covers patient for at least 9 months, but has the disadvantage of the may compromise islets engraftment. Basiliximab doesn't cause inflammatory reaction, but it's not the best as far as immunosuppression. So this is how we do it now, basically. So the Thymo first. So I bring the patients for thymoglobulin induction over 4 days. I can give them steroids. I'm not giving them islets, just to provide induction. Then I discharge patients, start oral anti-immunosuppression medication gradually, so patient adjusts to the dose to avoid side effects. And then after 3-4 weeks, I activate them on the waiting list. And as Trevor mentioned, in the United States, currently, there's only a few centers active, and we don't have any problems to find a really good donor and transplant those patients right after. Patient stays in the hospital for 2-3 days after the transplant. What we know that the islets need some time to engraft into the liver and recover. So we support patients after the transplant with the insulin and over the next at least 2 months. And then, but the clinical advantage is immediately. Patients typically are discharged on half of dose of the insulin, and it's much easier to control glucose from day 1. Still with insulin, but much easier. So based on our experience, if we use Thymo and tacrolimus and Myfortic, on average, 1 islet infusion covers 40 units of insulin. So if the patient is on 60 units of insulin, we're telling them most likely you will need 2 islets transplant, subsequent islets transplant, 2 or 3 islet transplant. If they're using more, maybe even 3. Now, based on Edmonton experience, we know that patient can get down the road even the 4th or the 5th transplant, and they tolerate it well. So this is just an example of the patient before the transplant. If you see poor glucose control, immediately after the transplant, the glucose control is much improved, but still taking some insulin. And after 2-3 months, we do mixed meal tolerance tests. We check the function, and either we stop the insulin or we offer them second islet transplant. So now about the regulation. So as I mentioned, in 2001, Edmonton Group, they introduced the protocol. And since then, and as you know, in Canada, it's reimbursed. It's reimbursed, and islet transplantation is recognized as alternative procedure and supported by the government. health systems in Canada, in Europe, in Australia, and Japan. And in all those countries, islets are regulated as tissue or organ for transplantation, not as a drug. In Edmonton, they did 700. I'm sorry, Trevor, how many did you guys done so far?
30, 40.
Per year?
Oh, no, 20 per year.
Per year, but overall?
Probably like 30, 40 total for the program.
Oh, I see. Yeah. Okay. So in Europe, they've done over 1,000 over the last 25 years. However, in the States, the moment in 2001, we wanted to introduce islet transplantation as any other procedure, FDA said, no, not so quick. Islets, we consider as a drug, and as any other drug, they need to be tested before they use. So the concept of how FDA see quality assurance for islets and for drugs. So basically, the thinking is if you use standardized material and if you introduce standardized manufacturing, you are creating a drug. The drug should have a standardized, consistent characteristics. So you should have always islets or the drug always with the same standardized quality. You can control it. This is the assumption. And then you test it in clinical trials, and then you have to show that the islet's characteristic, which is always the same, correlates with the clinical outcome. However, as we know that the quality of the islets is not always the same. We're just retrieving the islets. We're not creating them, at least the cadaveric islets. So the quality varies between the, despite standardizing the isolation. And then also the quality of the islets. We don't have really very sensitive tests to know if the islets are good or not. The same way as we don't know if the kidney is good or not. There's no single test. If somebody will bring us the kidney out of the blue, there's no way to say the kidney is good or not. It's kind of the same with the islets. But nevertheless, we have to follow that. And once it's proven that the islets as a drug are safe and effective, and all of it's proven, then the drug manufacturer has to apply for license and needs to validate all the procedures, the manufacturing procedures, and prepare the islets application. And this is extensive work and investment in financial investment. So what happened afterwards? So NIH, JDRF, they sponsor multi-center trials, and in those trials, we proved that the islets are safe and effective. But however, in the next step, the academic center is supposed to apply for a license like any other manufacturing company, and they didn't do it. We didn't do it. Our leaders of academic institution, they didn't want to invest millions of dollars in the license of the drug application. Our dean said, we are not drug manufacturer. We're not doing this. We don't want liability. We're not organized to run the drug manufacturing operation. And this happened in all academic centers in the States. And because of this, nobody had the license for over, once trials are finished, so over the next 10, 15 years. And because nobody has a license, insurance companies, they said, well, we can't pay for unproven drug. So there was no mechanism of reimbursement by the insurance all those years up to now, still not there. So the lack of funding, so there was no more research funding, and there was not yet the reimbursement. Therefore, the islet transplantation declined in the United States, and many centers, most of the centers, they closed the activity. So we, as the leaders in the field, we saw these shortcomings of the situation, and we proposed the solution. We proposed to adjust the regulations and let islets be regulated as organ or tissue like any other country, and it will allow islets to be reimbursed. We wrote several articles. We talked to FDA. However, FDA said absolutely not. We're not changing the regulation, and it's been over the last 6-7 years. So eventually, the leaders of the transplant community wrote a letter to the members of the Congress, inspired by us. And then one of the congressmen, Mike Lee, he introduced the islets bill in 2023, changing the regulations. However, 2 weeks later, FDA suddenly approved license for Celltrans, private company, for exclusive islets distribution in the United States. So because the license was approved, then Senator dropped the bill. So now we have the situation where Centrans owns exclusive rights for islets distribution, manufacturing in the United States. And for the last 2 years, they haven't opened the production. They haven't opened islets for transplant yet. However, University of Illinois, where they located, they advertising the program, and hopefully they will start doing some transplant. And those islets technically can be reimbursed. However, the problem is that the quality of the islets which Celtrans will provide, and they can provide to me, to anyone, cannot be really confirmed. We don't really know what type of islets we are getting. This is one thing, and then the Celtrans will decide which transplant center will do the transplant. They may offer to, they can select centers, they can offer islets. So it's not really available. It's not going to be available to everyone. And they will set the price. So the system, when there is single private company distributing islets, it's not really working in the States. So because of this, funding is limited. As I said, many centers there are closed down. We are lucky. We have some funding, ongoing funding, which allowing us to do the transplant. So 2024, we did 27 transplant. This year, we already did 15. So we are on similar trajectory. So what we do islets after kidney transplant, since those patients are on immunosuppression anyway. We do it. We don't have really funding for this. We have some residual funding from other trials. So this is very limited, but nevertheless, we try to help patients. So there are 3 main directions in islet transplantation, which we try to optimize. One of them is to find a more efficient site for islet transplantation than the liver. So Cernova is actually Canadian company located in London, Ontario, and they develop a pouch, hoping that it will provide better environment than the liver for islets engraftment. And we've been testing this. The funding came from Cernova and JDRF. We've been also testing new, less toxic immunosuppression, and I show you more. And then we also lucky to participate in Vertex trial when the stem cells derived islets are being tested. So those 3 trials give us enough funding to maintain the activity. UCSF has own funding, and they testing cadaveric islet transplant together with parathyroid stem cells, hoping that those stem cells, they have property to stimulate vascularization, and they've been testing this, injecting the islets into muscles. However, this is still ongoing and in development. City of Hope has own funding, and they testing intraportal islet transplant with gastrin. This is some examples of islets after kidney patients. So this was the patient with poor glucose control, high A1C, as you see. He got SPK, he thrombosed the pancreas right away, and he had peripheral vascular disease. We couldn't offer him a second pancreas. So eventually, once we get some funding, he got islet transplant. It has been with optimal glucose control for over 7 years. This is another patient. He received the living donor kidney. He had chronic hypotension on midodrine with peripheral vascular disease. So he got the living donor kidney, but continued to have poor glucose control. Unfortunately, we didn't have funding to help him with the islets at this point, and he lost both of his legs. He required a below knee amputation. Eventually, we gave him the islet transplant, and he's been off insulin. Another patient, SPK, she thrombosed the pancreas, and you see, more than 4 years later, we were able to get secure some funding and do the islet transplant. Another living donor patient got the islet transplant 6 years after the kidney, has been doing well over 4 years. This is our star. Joey was the very first patient at the University of Chicago who got islet transplant in 2005. She got 3 islets infusion, and she's still off insulin. She was on like 5 of Lantus. Now she stopped it completely. She started having hypoglycemia of 5 of Lantus. So we stopped the insulin completely, and she's 20 years later, still with the stable islet function. So we see islets has potential to work for many, many years. This is the patient reunion from the Dampet trial, which I mentioned before, right? So we needed some publicity, some fundraising. So we brought patients. At this time point, it was 2015, 13 out of 14 patients with off insulin. So we used this opportunity to promote the islets. So as you see, this was over 10 years ago. So I'm sure you're curious what's happening to those patients now. So those two patients, after one transplant, they've been off insulin for 10 years. And these are Reparixin patients. And these were BMI of 30. So this was something special about, in my mind, about Reparixin. One of them, the A1C recently went up to 6.5, so I just gave him a second transplant. These ladies, they over 10 years also after one islet transplant. And other patients, she was 5 years off insulin. Other patients were off insulin, but then they start requiring some insulin a few years later. So they came back. We didn't have funding, so we offered them whole pancreas transplant. And as much as they didn't want the whole pancreas transplant, once they tested life, enjoy life without insulin, without hesitation, they agree for pancreas transplant, and they still being off insulin. So going back to the Reparixin, in our cohort of patients, 50% of patients maintain at least 5 years insulin independence, which is really the best outcome in our settings. So this is one of those patients. So she was off insulin after one transplant for 5 years, and then she wanted to get pregnant. So we changed the Myfortic to Imuran, and then she went through 2 pregnancies on insulin. So we helped her with insulin during the pregnancy, and also protected the islets, because as we know, there is a lot of insulin resistance. And after 2 pregnancies and being a mother, now she's more than 10 years, and it's amazing. Islets function remains stable, almost the same mixed meal tolerance test over the years.
Just basal during the pregnancy or short?
I think both. They were targeting fasting glucose 90, and this was gynecology. So they were, look at this, and I let her go. I was not watching her. When I asked her, how much insulin you are on? She's like on 60. I'm like, 60? So islets are gone, most likely. But I checked C-peptide was really high. So she did 60 units of insulin on top of the islets to maintain the strict glucose control during the pregnancy. And afterwards, everything went down, and she's off. The side effects of immunosuppression, you know this stuff, so I'm not going to focus on this. We look at the neuropathy and the retinopathy of our patients over the years, and we saw what we suspected, right? That there is no progression of retinopathy or neuropathy, and in some patients, the changes are reversible. So now about the Cernova trial. So Cernova developed a pouch which we implant, and we decide to implant in the space between the rectus sheath and the rectus muscle, hoping that this is the best conditions for revascularization. Dr. Shapiro tried it in subcutaneous space, but there was seroma, there were a lot of problems. So we decided to go deeper. So in the protocol, we implant the cell pouch first. Then we wait 1 month for engraftment and revascularization of the pouch. Then we start immunosuppression, the timer, as I told you. Then we wait another month, and then we activate patient transplant. So patients getting 2 transplants into those pouches. And we knew this is a prototype, and it most likely not work. That's why I convinced the sponsor to pay for extra islet transplant in the liver, which is otherwise unavailable, so patient can get some clinical benefit. So there was extra transplant into the liver to let patients be insulin independent. In cohort A, which is 6 first patients, we found in 3 of 6 patients developed DSA. We saw some C-peptides, some islets survived, but half of the patients developed DSA, and I was mostly blaming patients that they're not compliant, most likely they don't take immunosuppression properly. So in cohort B, we decide to introduce IV immunosuppression, belatacept, to better protect and be more independent from the patient decisions. So in cohort B, the very first patient developed severe neutropenia, so severe that despite treatment, we had to completely stop immunosuppression. So the dose of belatacept and tacrolimus, which were introduced, most likely was too high. So we lowered it then, and then in subsequent, second patient actually was doing very well, has nice C-peptide, but during the second transplant in the pouch, on the culture after the transplant, we found a fungus. So unfortunately, it was telling us that possibly there is a fungus living in the pouch. So we decided to remove the pouch just to prevent this infection. Subsequent patients, we didn't find C-peptide, so it was a step back, and one of the patients developed DSA. So I couldn't understand what's going on until we saw the biopsy. So we explanted those sentinel pouches, and what we found, a massive CD3 lymphocyte infiltrates within the pouch around the islets. So what my conclusion from this is that the immunogenicity of the islets in this tissue, in this pouch, is much higher than immunogenicity of the islets in the liver. The regular immunosuppression, which is good for islets in the liver, is not sufficient for the islets in the pouch, and it's too much rejection, mostly maybe inflammation. So this is where we are moving forward, still trying to, maybe trying anti-CD40 ligand immunosuppression, trying more optimal immunosuppression. But at the same time, I think it would be really beneficial to try stem cells-derived islets, which are less immunogenic. They can survive better in different environments.
Oh, just not immune.
No, it's just supposed to provide better engraftment and vascularization. And this is the example. This is one of the first patients who had C-peptide for a long time, over 2 years, and then we transplant her into the liver. But she decided to keep the pouch for 5 years, and we explanted, and we see nice islets, nice vascularization, but it's still not sufficient to get patients off insulin. They need more. They need something more. All right. So now about the minimizing the toxicity. So yes, we are very fortunate that we've been having chance to test anti-CD40 ligand antibody. It's a tegoprubart developed by Eledon. Just to give you some background information about this drug. This drug was developed 25 years ago, and in the monkey and in the pig, it was great. It was very effective. In monkey, in islet transplantation, islets were going only on this drug, on one drug. And when after half a year, they stopped the drug, the islets were going. So this was really exciting. We hope that this is the drug which can stimulate tolerance. Then the first clinical trials in kidney transplantation, patient developed thromboembolic events. And this stopped the whole trial because this antibody was stimulating thrombosis, and the drug disappeared for 25 years. And this is how long it took the company to modify the molecule so it's not prothrombotic anymore. And now we're testing this in clinical settings. So there is a multi-center kidney trial now, standard of care with tacrolimus versus tegoprubart in combination with the CellCept Myfortic. So completely tacrolimus-free immunosuppression, and the results are promising. Seeing those results, we were excited, and we get some funding from JDRF, Breakthrough T1D, and the Cure Alliance to test this immunosuppression on first 3 patients. And the results are good. I'm going to show you in a moment. And now we get more funding to do 6 more patients. In the meantime, without much funding on my own, I've been testing the belatacept with low tacrolimus, adjusting the dose, and seems it's working well now. Once I figured out the optimal dose. So this is the schema for this Eledon trial. based on the kidney experience, in kidney transplant, they saw a bit more BK. So it's telling me there was a bit, the dose was designed maybe too high. So we decided to go with lower TIMO, provide a bit less immunosuppression. So the dose is 3 times 1.5. Then we wait 1 month and then we go with the transplant. The goprubart around the transplant is given on day -1, 0, 1, 3, and 7, then every week for 1 month. And then patient comes for 1 hour infusion every 3 weeks. And my Fortic, we start with 720 twice a day, but we go down as soon as I see some CMV activation, some positive PCR, I go down with the dose, and then CMV PCR disappears without any clinical symptoms. I just feel comfortable that we can lower the dose. So these are the very first 3 patients. So the patient number 1, high BMI 30, and the patient was on, okay, we gave them 360,000, which is average. It's not the highest number of islets which we can find or get. So around 350, 320, 370. So average number of islets. And then in each patient, decrease insulin requirements at least 60 units per day. The very first patient, she required 80. She went down to 16 per day after 1 transplant. Second patient was on 60. She became off insulin 1 month after the transplant, and she remains off insulin now, 10 months later, with A1C of 5. And the third patient, he was on 90 units of insulin and decreased to 16 after 1 transplant. So we see that it seems to be more beneficial than tacrolimus. As I told you, we usually see it, the decrease, 1 transplant decrease over 40 units. So we look deeper in this, and we look at the islets engraftment again, based on the mixed meal tolerance test and C-peptide. And then, so this is with tacrolimus and entanercept. This is my best patients with reparixin engraftment, and this is the engraftment with this new immunosuppression. So it's even better than reparixin. It's very exciting. And I think one of the elements which we never appreciate, we know that tacrolimus is diabetogenic. It's toxic to the islets. But only by removing the tacrolimus, we have such improvement. It's really something exciting.
His face.
Sorry?
His face and those 3 patients.
No, 0. No, nothing. No DSA. They're doing this rock stable so far.
And no trunk.
No thrombosis. No, it's not in the kidney trials. And the kidney, they have 60 patients. And no, not really. No.
And CMVs.
So again, I'm very sensitive about this, knowing about the BK. So I'm watching EBV PCR and CMV PCR. And as soon as my patient develop like less than 35, which is the first step, I lower this, I lower my Fortic, and it's gone. So none of them has any infection. And then, you know, I don't do any adjustment. Like we always adjust the tacro up, this does creatinine up, this white count up, right? We all constantly adjusting medication. Here, there's no adjustment. I just see them every 3 weeks. We shake hands, and they're doing so well. So this will change the way we work with the patient if it's working. Of course, something may happen, but so far so good. So now shortly about the stem cells derived islets. As you know, Vertex has technology to manufacture human islet cells. And we've been testing this, and Trevor was a leader for a long time, but now we did a few more. So the Vertex 880 trial, we transplant the islets into the portal vein, into the liver, the same way as we do with the cadaveric islets, and this trial is ongoing. And looks like those cells are really working. And nothing bad happened to the patient because of the cells. In the parallel, Vertex organized trial 264. They put the same stem cell-derived islets, but in the immunoprotective pouch in abdominal wall. Unfortunately, the pouch doesn't work. There was no C-peptide observed, and the trial was stopped. ViaCyte has been working on the stem cells, which they genetically modify to be hypoimmune. I haven't seen any clinical results yet. So yeah, so this is the Vertex. The beauty of this is that we schedule the procedure, they ship the islets over the night, and we just hang the bag. And those islets are working. So I think this is the future. The question, I mean, hopefully nothing will happen, and then this will be licensed and available. Now it's available only from blood type A or A. I mean, the donor is A, so it can be A or AB. But the company, they will have the cell lines for every blood type. So these were the results from the last year ADA. In a few weeks, we have ADA in Chicago. There will be much more patients. But the bottom line is islets do work. A1C goes down. Patients are stopping insulin between 6 and 9 months. So it takes longer for those cells to adjust, to engraft, and most likely mature to have full potential and have patients of insulin. So between 6 and 9 months, this is where we typically see patients are able to stop the insulin afterward transplant. But it just occurred to me that the Vertex trial limits the insulin usage for patients. It has to be below 60. So they set up the trials to have one transplant sufficient for this. But anyway, as I told you, we have possibility to do the second transplant with Vertex cells if necessary, assuming patient. And what's also interesting is that none of the patients so far, they're saying had any DSA on oral immunosuppression. So to me, it's saying this, it's not a perfect word. We know patients are not perfect, right? So potentially the immunogenicity of the stem cells islets is lower since none of the patients develop any DSA on oral medication. So I'm sure you heard recently about these results from China. So in China, they developed technology, amazing technology, if it's true and can work. They taking fat, isolate stem cells, convert them into the islets, and they infuse them in the rectus muscle and own islets from own stem cells, and patient is insulin-free. They use a patient with the liver transplant on immunosuppression, but nevertheless, the idea is that these are own stem cells derived islets. So this needs to be verified. And I know the primary investigator is coming to WTC as an invited speaker, so he may show more data. And then Sana is the company. They develop hypoimmune islets by genetic modification. What they did, they knock out the HLA, class 1 and class 2 on islets, and they upregulate anti-CD40 to make them protected from NK cells. They show one monkey, and it did work, protect the islets, modify islets without immunosuppression. And now they tested this in first patient in Sweden. However, they test the concept. So what it means, they transplanted small amount of islets into muscle, and they just measured the C-peptide, no immunosuppression. And then what they're showing is there was no C-peptide before. Patient maintains, it's a teeny tiny amount of the C-peptide, but they put small amount of islets. It's hard to say. This is what we have, and I think they also said they see the same C-peptide now after 3 months. So there is something here. But what's interesting here, when I look into details, the way they design, I think it's excellent design, because what they did, they put some islets which are modified among normal islets, unmodified islets. And they put in the patient without immunosuppression, allogeneic islets, right? So patient did develop rejection to unmodified islets. There was donor-specific antibodies, and despite that, those modified islets, they see C-peptide, they're saying they survive. right? So this was experiment to show that despite all this rejection mechanism, those cells can survive. It's small amount of C peptide. It's one patient we need to see, but this is very exciting. Oh, and we are about to start the new Vertex trial with the same way, just in the patients who had already kidney transplant. So they will have chance to help those patients. So we have website with our patient stories to promote the islets and tell everyone that the islets can work. And we still hope, I'm losing the hope on changing the regulation in the States, but we have now new FDA leader, and he is a surgeon who was the islets director in Johns Hopkins for auto transplant. So he knows islets. He knows islets. He hasn't responded my emails yet, so I'm not holding my head. I mean, it's up and down, but this is the last moment. If he doesn't change the regulations, nobody will change it. Thank you very much.
We're happy to take any questions from the audience. We can also look online if there's any questions. Omar.
I mean, thanks, fantastic results, and especially having in mind how difficult the environment in the US is, so that you're keeping it up and bringing this innovation forward is really remarkable. If we come back to the long term, you know, after Edmonton, everyone was excited, and then the 5 and 10-year data came out. And although we know that insulin independence is not everything for the patients, it's very important. And they had like 10% 10-year insulin independence, while with pancreas, it's like 60%. And so in your experience over the years, and you modified the immune protocol, you went away from mTOR, more MMF-based. So what's your 5-year insulin independence with your recent protocols?
So I think what the key is the initial engraftment. The higher it is, the higher chance the islets will go for a long time. If we put marginal number of islets, gradually they will go down, regardless of immunosuppression. So the key is load the patients with this. That's why the Parixin patient, we had bigger, higher engraftment. They went so well over 10 years without on one transplant, right? Because they get more islets at the very beginning. I think this is the key. When we giving, one transplant or 2 transplant, and we are at the borderline, just to have patients off insulin for now, but this is suboptimal islet mass, they gradually going down. So the key is to give enough islets at the beginning, and then the maintenance, right? Again, I don't think time was important to protect at the beginning or something, right? So now I do believe that to be more independent from patients on decisions is belatacept is the way to go before the anti-CD40 is available. So I'm giving now, I switch all the patients to belatacept to 5, and after 1 year to only 2.5 per month with the low tacrolimus. And so far they're going well. I can't give you the long-term outcomes, but I do believe that this is optimal for now because it's not toxic. Tacro level is only 3, and having this IV, it's just protecting them well, even if they seek vomiting and stuff like that. But I think the key is the initial engraftment, to give as many islets as possible, and allow them to survive as many islets as possible.
See.
Fantastic talk, very informative. I was wondering about your comments on the Sernova results. Do you have any idea whether the infiltration that you see is due to the location, or is it due to the pouch itself? I mean, has it been done without cells?
I hear you. I didn't test this, but they tested before. My understanding is they didn't see the infiltrates or foreign body reaction to the pouches in itself. We do this sentinel pouch removal after the islet. So I didn't really go back and look if there are infiltrates with the pouch itself, but I think it's a great idea, to look into this. Just to make sure, go to the basics, right? Right, yeah.
Just curious if we talked a little bit about GLP-1 inhibitors.
Right.
Do you guys use any of the other sort of augmenting drugs for type 2, like SGLT2?
No, I believe it's beneficial, especially for those patients who are obese, right? Who BMI is high. And before, when GLP started, Jose Oberholzer, he showed the benefits of GLP-1 when the patient had like borderline function going down. The GLP-1 is helping them. So definitely there is a beneficial role. If, I can't have it in the trial now, but moving on like you, I would give it to the patient. I mean, as long as they tolerate, as long as they kind of obese, BMI over 28, I guess, to decrease the insulin resistance and help patients losing weight or limited food intake, right? And maybe help islets as well.
Yeah.
Problem is it's not covered for type 1.
Right. No, so then I'm telling them, no, they type 2 now, because they have nice C-peptide, right? And A1C is 6.5, right? So they type 2. Sorry.
Very good.
Thanks for a very interesting talk. You mentioned the abundance of donors. I'm wondering about HLA matching and whether
Right.
effort in that regard.
Yeah, so I can tell you. So one of the advantage now of this poor situation is that we have entire country and we are in the center. I can bring the organ from California, Seattle, Miami, all the country. So I have patients with PRA of 95, and I can find donors without any DSA. And they're coming, so they wait half a year. So this is the advantage for now. So yes, we do. We screen the patients for any HLA antibodies, and we're choosing the donor. Even the historic ones, even if they don't have them now, I'm just excluding those donors to prevent memory response.
And are you trying to HLA match the organs themselves?
We didn't see this as an advantage, so we only match the antigens and antibodies present in the patient. Because theoretically, we are afraid if you match too much, there might be autoimmune recurrence. But if with the stable immunosuppression, if there's 6 antigen mismatch, they still go.
I was going to ask about the autoimmune part because I always worry about that with these patients. So are you testing for anti-GAD before or after transplant?
We've been doing this. It's expensive. The test is $500. We've been doing this for a long time, and we didn't find any correlation. The patients, there's one of those two, he has up and down of GADs and stable islet function. So it's not black and white. So it's not really useful.
Every organ we transplant has an acute rejection rate from 10 to 25, 30% in the first months.
You mean like pancreas, kidney?
No, like kidney, pancreas, livers, whatever we transplant.
Right, yeah. Okay.
In islet, we don't recognize it because we cannot really do biopsies.
Right.
So, but assuming that it's the same, there were suggestions that just by metabolic control, you could basically say, well, this must be, if everything else is included, rejection. Right. Do you screen for something like that?
So I think.
And do you treat acute rejection?
Right.
Yes.
Let me tell you. So I think this is what was happening with basiliximab in initial experience. It was enough to prevent the DSA, but most likely the cellular rejection was not controlled sufficiently. That's why the patient were losing. So I can't do much more than monitoring DSA, right? I can monitor the cellular component. So my goal is to cover them as best as possible at the first 9 months, right? That's why idea about belatacept, it's really resonates with me that I'm giving and I know they're getting what they need to protect this rejection as much as possible. Now, we had the patient, one of those ladies, she developed, so she went home after a week, she was kind of fine. And then after 1 month, she called me, oh, I need more insulin. So we brought her back and she had 90% PRA, she had DSA. So we did plasmapheresis, IVIG, rituximab, and all DSA went down, 0 PRA. I was able to remove them with plasmapheresis, 5 plasmapheresis. And then we gave her second transplant, avoiding those DSA, and she's 10 years now after. So we were able to save her, right? And she maintained the C-peptide of the first transplant. So she did respond to plasmapheresis. There was a second patient who developed acute rejection, as you actually mentioned, on day 7. This was one of the very first patients with the cytokine release, right? So I do believe the cytokine release stimulates rejection as well. So he developed PRA of 90 with DSA on day 7. So we start doing plasmapheresis on him and this, but C-peptide disappeared. It was too late, but at least PRA went to 0. And then we transplant him second time. He was off insulin. But the third patient developed DSA after 2 weeks. We plasmapherese him 5 times. And after every plasmapheresis, DSA is the same. I couldn't even believe that they were doing this. It was exactly the same. We couldn't overcome it. So I just gave him the second IV, the big dose of IVIG. And then he maintained some function, but then we gave him a second transplant. And then we gave him a third transplant. So what happened with the time that the DSA against first one, they decreased to 0. Most likely he lost the first cycle. But we were not able to remove the DSA. So some patient it did work, some patient.
Other questions?
One more.
I have a quick question. I just wondered if you could comment on the FDA's decision to make islets a
Right.
biological product, or what's their resistance to changing it?
Okay, so they decided about islets being a drug in 1995. And then they continued the same decision in 2001. And now they're saying, we have to stick to our decision. This is the law. We hear you, but no. Now they say when we close doors, right? When I talked to Peter Marks, the CBER director, he's like, if we do it for you, then others will come with different therapies and they won't do the same. We can't do exceptions. So in his mind, this was a big picture. They don't want to create a situation. But in our transplant situation, vascularized like hand transplants and stuff. So tissue transplant and hand transplant, they were regulated by FDA. And the decision was changed by the Secretary of HHS to include them under organ transplant. So in transplant, such decision was made and can be made. Only this is decision of the Secretary of HHS. By the law, it's his decision what's organ, what's not organ, and he can change it. So we try with HHS, but whenever we ask HHS, the answer came from FDA. On behalf of HHS, we told you no. So it's a circle.
Thank you. It's a great talk.